was like the reoccurring host that we kept finding Phytophthora tentaculata on. I think, I also made the statement that many Phytophthora species are a problem in nurseries, and I was making this based on our knowledge from ornamental landscape nurseries. Up until that point in time, we really didn't have much information on what was going on in native plant nurseries. And I think the main message that I talked about, and many others, was that the worries about the spread of Phytophthora tentaculata and other Phytophthora species in nurseries, and then the subsequent damage that you can see in the wildlands, like Susan talked about. So you guys heard that message and you responded. We had a large influx in calls and interest in people wanting to get their native plant tested by our lab for Phytophthora species. So up until the Presidio meeting, we had done about 300 samples for Phytophthora root rot testing. Since that time, we've done over 2,000 samples for Phytophthora. And this includes nursery-grown container plants. It includes outplanted material. We receive plants and roots, as you can see here. We also received baited pairs, where people out in the field were doing their own baiting from water, from baiting from root balls, from flow-through. So we would receive those pairs to do Phytophthora testing on. And we also received cultures from people who were doing their own culturing, like Liz and Ted. And they would send us the samples for Phytophthora ID using molecular techniques. So just kind of a snapshot of the nursery samples. And I'm just going to talk about here just the samples that we looked at where we looked specifically at the roots themselves, what we could culture and identify from the roots, not baits or flow-throughs. Through January of 2016, we did about 400 root rot samples. This included samples from 26 nurseries in 14 different California counties. Over 100 different plant hosts were sampled and sent to us, and these were collected either by CDFA people, extension, and also just private nursery owners. We used ELISA culturing PCR sequencing for identification. So from this group of samples, we detected one or more Phytophthora species from about 37% of the nursery samples. The most commonly detected species was Phytophthora tentaculata, and I don't think this is really completely accurate in making the generalization that tentaculata is the most common species in native plant nurseries. I think it was just heavily sampled in these by the nursery owners because we knew the hosts of Phytophthora tentaculata, and those were the ones that people were concerned about. So we had tons of diplicates that were sent to us. So it might be a little skewed in regards to that one. It was followed by Phytophthora cactorum or cactorum-like isolates, species in the Phytophthora cryptogea complex, Nicotianae, Niederhauseri, Hedrondra, and so on down the line to less commonly detected species. And I imagine Tyler will talk in more detail about these different species. So overall, we had about 13 different species that we found from this group of samples. So of those 26 nurseries that sent us material, 20 of them we found Phytophthora in. A few of them that sent samples, they only sent maybe 2 samples, so I think it's hard to say that they don't have Phytophthora. Maybe we just didn't have enough samples or they weren't the right samples. 58 of the 102 hosts that we sampled were positive for one or more species of Phytophthora, and these are all the hosts that we had Phytophthora detections from. The number in parentheses is the number of Phytophthora detections. I do have like a large table showing all the species that are associated with these, but it was just a giant table that would be hard to read and understand, so I just simplified it here. The species in red are those Phytophthora species that we detected Phytophthora tentaculata from. So not only is regular sticky monkey flower, the orange one, Diplicus aurantiacus, susceptible, but many of the other varieties are as well. Artemisia species look to be quite susceptible to tentaculata. We found it on 4 species. We've also found it on Ceanothus cuneatus and Monodella villosa. An interesting thing here is look at all the species on Arctostaphylos. For example, Arctostaphylos pumila, We had 12 different Phytophthora detections from them. In one lot of pumila, we had 4 different species. So kind of interesting results. So overall, we found that in addition to being common in commercial nurseries, they are common in native plant nurseries. And as an example, in one nursery that we intensely looked at, I think we had like 80 samples from this nursery, 60% of those that they sent us were positive for Phytophthora, and that included 34 different plant hosts, and 12 of the 13 species we found were in this one nursery alone. So quite kind of shocking to find so many in one location. But it also shows that really once you start looking, the more you look, the more you'll find. And these are some examples of Phytophthora species infecting roots of some of the different hosts. We found many new Phytophthora host associations, which you saw on the slide, previous 2 slides. We also found a Phytophthora species that wasn't previously known in California. Phytophthora parviflora actually wasn't known in the US before. It was found on Choisia tornata, and we found it causing a root and crown rot, as you can see in the left side. And this species is known in Europe, South Africa, where it causes crown and root rot on mandevilla, citrus, and the picture on the right is symptoms of it causing root, crown, and dieback on Arbutus unido in Sardinia, Italy. So to summarize, Phytophthora tentaculata's host range has really expanded. We've gone from 4 hosts to 10 hosts, from 5 to 8 counties in California. And though the concern really started with tentaculata, it's really evolved into a much bigger issue in regards to all these Phytophthora species that we really didn't know much about. Phytophthora species are a problem in both ornamental and native plant nurseries. In nearly all the nurseries that have sent us samples, we have found Phytophthora. We also know a lot more about native plants and really their susceptibility to Phytophthora species. Sticky monkey flower plant, Diplochus aurantiacus. Prior to this work, really, there was no reports of Phytophthora species on this host, and now we know there are at least 10 Phytophthora species associated with it. So it seems to be, it's like one of those hosts that if a Phytophthora looks at it, it seems to get infected, so very susceptible. We also know now that host pathogen interaction is quite important. Some species, like Diplochus, are more susceptible and get more Phytophthora species. And other species like Toyon, they do get Phytophthora, but they tend to be a single host. It's usually Cactorum. So, and we also know that some hosts that we once thought were unlikely hosts can also get Phytophthora, like we've seen it on grasses and monocots, for example. The diversity of Phytophthora species varies among nurseries, and this depends on climate, host species, and all the different type of management practices that are used in those nurseries. So I think it's been very interesting to see the different practices that people have, BMPs that people have implemented, and starting to see differences. And I'm really interested in what we're going to learn in the next 2 years. We've learned a lot in the last 2 years, and so interested to see what we'll learn in the future. And just a final slide I'll leave you on, just to kind of show you, this is Diplochus, kind of how I like to show this slide. Some people have probably already seen it, just to show you really how hard it is to determine, to judge a plant just by its above ground symptoms. Can you tell which ones are infected with Phytophthora tentaculata or other Phytophthora or are healthy? Well, I put them in order of what I thought, and actually the healthiest one had Phytophthora tentaculata and the worst one. But the kind of the second worst one, I thought, I didn't detect anything from the roots. And then 2 of them had cryptogyia. So just a kind of don't judge a book by its cover. So with that, I thank you for your attention.